Cutter is among us for delivering his talk. And as you can see, he is expert in base editing and prime editing. And recently, he has developed a new method of genome editing and published his paper in very reputed and high impact journal, Plant Biotechnology Journal. So we are lucky that Dr. Qutub is here with us today. So I invite Dr. Qutub for his talk, please.
स्लाइड मूव नहीं कर रहा है. थैंक यू. आपने खाली इसी को नहीं जी इसको थैंक यू. इससे भी हो जाएगा ठीक है. Okay, good morning everyone. Thank you. Thank you Dr. Anil Kumar Singh, Principal Scientist in IPB, and as we met I think last in Agri Science Congress, sir. Thank you for your invitation and giving me a chance to talk to your trainees. And I'd love to see all of you together. If some are sitting in the back, if you could come forward, I would be encouraged to do more, better. Okay, thank you. So I think how many days is over in this training already? Fifth day. So fifth day means you already know what is CRISPR-Cas9, how does
Recording
it work,
in progress.
and how does it work, and how also experimentation could be done and should be done. So I think you are also going through some practical hands-on experience, as I have learned from the course coordinator in the afternoon. So today I take you to some advanced technologies. Oh, this was muted. Okay. So I will be taking you to some advanced technologies. These two technologies named as base editing and prime editing. But what we can do with this technology, because a lot of hype in the news and all news house, they did a lot of hype on CRISPR-Cas9 that it can do anything, but it cannot do anything. Conventional CRISPR-Cas9 or canonical CRISPR-Cas9 cannot do everything that you want to achieve for modifying your genome. So that there is a lot of limitations, and I would be talking about two of the technology, some of the limitations of canonical CRISPR. opposite strand it is G to A. So one strand, if it is changed C to T, opposite will be automatically changed to a G to A. Now, but base editing has limitation. What is the limitation? It cannot edit any base of your interest if it is not present in an optimum activity window. What does it mean, optimum activity window? Here is a 20 base pair protospacer of the guide RNA. Suppose, for example, this is the genomic sequences where 20 base pair target and then followed by PAM sequence, NGG. But the base of your target base should be located between 4 to 8. Then only this base editor would be able to optimally change your target base. Got my point? So this 4 to 8 region, this span is called activity window of base editor. If your target is present at 11th position or 12th position, it may not act as optimally as it was acting a target which is present at 5th position or 6th position. Am I clear? If I go too fast, please raise your hand. हाँ ठीक है तो मेरा objective of this lecture is to make you understand about this technology, to bring your interest to read more. That is my objective. Okay, अगर इस objective से बाहर आप जा रहे हो तो आप मुझे खाली हाथ उठाओ, फिर मैं रुक जाऊंगा, आपको समझाऊंगा, फिर मैं आगे बढ़ूंगा. ठीक है तो clear? So optimum activity window समझ में आ गया है. So how many proteins are there for cytosine base editor? N-Cas9 is a deaminase, and this is another orange protein. Just remember orange protein. Okay, so how it actually works? Let's see here.
Then be used to transport those enzymes to a specific DNA sequence.
So this orange is deaminase here.
In one example,
And
Cas9 is
blue
fused to
is
an
Cas9.
enzyme, a deaminase, which mutates specific DNA bases, eventually replacing cytidine with thymidine. This kind of precise gene editing means you could turn a disease-causing mutation into a healthy version of the gene.
Okay. Now, how it actually happens? Is it only a story, and what is the chemistry behind it in the cellular environment? Suppose you have a defective gene which has a defective base pair CG. You want it to be corrected as TA. Okay. So what base editor you would be using? Cytosine base editor. So first you apply cytosine base editor. As cytosine base editor has a deaminase protein, this deaminase will deaminate cytosine into uracil. If cytosine is deaminated, it will form uracil. Now it becomes a UG base pair. UG base pair is a mismatch. It is not a true match. U never pairs with G. Have you heard about that? U किसके साथ करता है? A के साथ. So it's a mismatch. Now cellular mismatch repair would come into action, and it will immediately try to figure out which one is mismatch. U is mismatch or G is mismatch. Now क्या आंसर होगा? Cellular mismatch repair machinery trying to understand. उनको क्या पता होगा? U खराब है, G खराब है. So उनको पता नहीं होगा. They can remove U, they can remove G, either one of them. So in order to trick the machinery, this CBE is made up of N-Cas9. I told you N-Cas9 means it can make only one single nick. The N-Cas9 will make a nick at the G containing strand. अगर उसमें निक हो गया, सेल को अभी पता चलेगा G गलत है, क्योंकि इसी स्ट्रैंड में निक है, ऑलरेडी डैमेज स्ट्रैंड है. So G is the wrong letter. So G would be removed, and opposite to U, A would be inserted, and then in the next round of replication and repair, it will be TA. So this is how CG becomes a TA. But in normal higher eukaryotes, it's not that easy. There is another protein which inhibits this pathway. The protein is uracil glycosylase. Why? This is a base excision repair protein. Uracil glycosylase. क्यों आप बोलिए तो U कभी DNA में होता है? नहीं होता है. गलती से अगर U इंसर्ट हो गया तो सेल उसको तुरंत जीनोम से निकाल देगा, जीनोमिक सीक्वेंस से, क्योंकि U आपका जाएगा RNA में है, आप DNA में क्यों आया? ठीक है, तो यूरासिल ग्लाइकोसाइलेज उसको निकालने की कोशिश करेगा. एंड देन so this outcome is prohibited by the highly active uracil glycosylase. Now comes the orange protein's action, uracil glycosylase inhibitor. It will inhibit uracil glycosylase from removing U from the genome. So the lifespan of U now is increased because of the inhibition of uracil glycosylase, so that you get this outcome, and this is how the whole CBE is made. Okay, I am not spending because I have a lot of story to tell you. There is another class, this is a famous class, is more precise, even adenine base editor that causes A to G in one strand, and in opposite strand it causes T to what? C. Clear? Now it is made up of only two. One is N-Cas9 fused with a adenosine deaminase, and it has also different kind of activity window. One version has an activity window ranging from 4 to 8. Another version also has a little bit extended activity window, 4 to 10. Clear? Now in this case there is no extra orange protein required, because the here once adenosine deaminase deaminate adenosine into inosine, it becomes IT base pair. Then IT is also a mismatch, because I in the cell it is read as a G. I in the cell it is read as a G. So G किसके साथ प्यार करना चाहिए? C के साथ. So that's why IT is also a mismatch. And then the similar mechanism, NAHEP-CAAST will make a NIC at T containing strand, T would be removed and opposite to IC would be inserted, and then you get G2C. So AT become GC. But here there is no additional protein needed as the CBE, because there is no active inosine excision repair mechanism in eukaryotic cell. So there is no inhibitor of this outcome. That's why there is no need of using another protein. Okay, let us go to the real example. Okay, those are all technical details, but sometimes technical becomes boring. But this is also interesting to know how it happens actually in the cell. So this is one real example I am showing here. The gene is white stripe leaf, WSL5, and can you just count here how many base pair is here? It's 20 base pair, and this is the PAM in the opposite strand. Opposite strand में PAM किधर होना चाहिए? अगर आपका ऊपर में, if your protospacer or 20 base pair is present here, the PAM should be here, NGG, yes or no? But if it is in the opposite strand, then you have to count from this direction, okay? 5 prime to 3 prime direction. So 20 and then 3 base pair, NGG, okay? PAM. Now our target is this target A, this A is our target. And in the literature it is known that if this A is changed or modified, then the plant would look like a white stripe leaf. It will not be completely green, it will have white stripe. This is already known in the literature, it's a natural mutant described. And the A is present at what position in the protospacer? Is it in the activity window? Please count from this direction, highlighted yellow color. The target A is this one. कितना number position में है? What position? 5th position. So 5th is within activity window. So now, so it is good to edit. It's a good target, okay? Now another one is Jevro 3. Now can you count also similarly? Here is NGG PAM and this is protospacer. And our target is this A. And what is the position of this A? 7th position. 7th is also within the activity window. Now another thing I would like to just pay attention to, this A is also falls within a restriction site. Which restriction site? Blue highlighted? SAL1. The previous one, it is within a SAC1 site. So now why we have selected this two target? If your base editing is working, then you can easily detect them by only digesting your PCR product. If it is being digested, base editing did not work. If it is not being digested, it means A has been changed, and that's why the restriction recognition site is disrupted and you are not getting any restriction digestion. Okay, so we have first done in protoplast, and you see here this is wild type undigested without any SAC1 treatment. With SAC1 treatment completely digested, and you can see these are a bit different samples of protoplast, and you see lot of RE resistant band, which is an indication of what? RE resistant band is indication of base editing. for the first gene WSL5. For the second gene also, these are RE resistant band, which is also an indication of simultaneous editing in the second target. And this is a protoplast result, only like 24% editing for WSL5 gene and some 14% editing for Z3. But while we have regenerated the plant, now getting excited from that result, then we transform them for a stable transformation, we regenerated plant, and we have seen complete changes. This target A is modified to a target G. You can see green peak is modified. No, it's a fresh callus transformation. Protoplast is for indicative experiment we have done. Okay, now we have faith or confidence on our vector. Now we did the same transformation into callus, and now this is regenerated plant, and you can see this is the control chromatogram where the green peak means A. All of you know, no? Green means A, red means T, black means G, and blue means C. These are the four color of how to read Sanger chromatogram. So this green is changed to a black peak. It means A has been changed to a G nucleotide. Very precisely, no other changes has happened. And as a result of these changes, biallelic changes, you can see the expected mutation also we got, that is white stripe leaf. For the second target also, because we targeted both of the genes through a multiplex base editing experiment where the same base editor is present, but we have given two different guide RNAs, one for Z3, one for WSL5. So the base editor goes to both the target and it performed both the editing. So here the 7th position, 1, 2, 3, 4, 5, 6, 7, 7th A has been modified to a G nucleotide as expected or as desired. And you can see this is the mutant phenotype as expected. This is zebra 3 mutant, means light green, dark green patches in the leaf. Okay, so it means the vector is really working very precisely for base editing. Now this is in my lab. So what we are doing, the first one that I have already described, that I have done in my own hand, and this is done by my student. So kinetic and structural data suggests that there are a lot of modifications, huge kind of rapid, is more than the Vande Bharat speed this is going. Okay, this this field actually. So ABE80 is a new kind of deaminase that has been discovered from the same pioneer group, David Liu, which is 1100-fold faster than earlier ABEs. So we try to develop a similar kind of base editor using this highly efficient deaminase. And you can see here this example. This is the earlier version of ABE that I have just described, ABE 7.10, and this is ABE80 NGG and ABE80 NG. Can you tell me what is NGG and NG? Anybody, if you are exposed to already? NGG NG क्या है? NGG क्या है? NGG तो आपको पता होगा क्या है. कभी कभी CSR एग्ज़ाम में या कोई भी कॉम्पिटिटिव एग्ज़ाम में बहुत कुछ पता नहीं होता है, आपको गेस करके आंसर देना पड़ता है. आपको तो एक चीज़ पता है, NGG पता है, सिमिलर जगह पे NG है, इसका मतलब क्या है? मतलब और एक PAM होगा. NGG is a PAM, this is a normal Cas9, but there are some Cas9 variant or engineered Cas9 that can recognize NG PAM, not NGG. Sometimes NGG PAM availability is a limitation, but if you do not have NGG PAM, you can use this version if you have only one G. NG, N means any nucleotide. So presence of a single G is enough for you to target that gene. Okay. So this is an engineered Cas9 version, N-Cas9 version, and this is a wild type. So both of the base editor we have tried, and you can see just you compare the RE resistant band. What is here? Very faint band is there. It means editing happened, but faint. Here NGG version is the thickest band, and then a little lighter band with ABE80 NG. Can you tell me what are the performance of this 3 base editor? Which one is the most potent base editor? ABE80 NGG is the most strongest base editor. Then ABE80 NG, and then ABE 7.10, that is also NGG version, ABE 7.10. Okay, so you can see the 4 differences in their activity in protoplast. Now remember one thing I should also mention to you, because many of you probably go back and do experiment. Whatever you see in protoplast efficiency, that is likely to be increased in in your stable transformation. Why? Because we normally do protoplast transformation and harvest the DNA within 3 days, 72 hours. 72 hours, and there is no cell division of protoplast. If you incubate 72 hours a protoplast will not divide. We do not give them any kind of media that will encourage them to divide. So इसीलिए कोई भी जीनोम एडिटिंग एक्सपेरिमेंट में जितना सेल डिवीजन होगा आपका एफिशिएंसी उतना बढ़ने का चांस है. सो आप अगर सेम एक्सपेरिमेंट कैलस में करते हो, प्रोटोप्लास्ट में आपको जो एफिशिएंसी आया कैलस में उससे ज़्यादा आएगा. क्यों? कैलस इज ह्यूजली डिवाइडिंग सेल, 1 से 2, 2 से 4 ऐसे हो रहा है ना, पूरा छोटा से बड़ा हो रहा है. सो इट इज़ अ डिवाइडिंग सेल, सो यू हैव ए बेटर चांस ऑफ गेटिंग मोर एडिटिंग. Okay, so when we did Sanger sequencing, let us see the performance of PKAV80. The same target, you remember, all the Z3 our target was 7th position G last time, याद है? Z3 में 7th position A हमने target किया था. Yes, A has been modified to G, the black nucleotide. Now what you see in the 12th position there is A. That A also has been modified. Now tell me what is the reason here. आपको पहले 8 के अंदर हो रहा था, अभी 12 में भी हो गया, very efficiently हो गया. Almost all sequencing we have done, everywhere we have seen the 12th A also has been modified. It means it is very strong and its activity window is expanded or longer than the normal AB 7.10. Okay. Now second one also we have targeted, and in last time I have shown you in my chromatogram that only 5th was changed, but this time 11th A was also changed in the first target WSL5. So it means it has really expanded activity window. So now it has also a problem. What is your problem? Problem is, suppose your target is only the 5th. You do not want to mess up with the 12th or 11th, but you can get editing of that one also. सो एक्सपैंडेड एडिटिंग विंडो होने से थोड़ा सा प्रॉब्लम है. सो आपको जितना स्ट्रांग होगा, स्ट्रांग परफॉर्मर का भी प्रॉब्लम होता है. दिस इज अ स्ट्रांग परफॉर्मेंस प्रॉब्लम. ओके, एंड दिस कॉल्ड बाई स्टैंडर्ड एडिटिंग. बाई स्टैंडर्ड मीन्स आपको डिजायर्ड चेंजेस जो है उससे ज़्यादा ये चेंज कर रहा है. ओके, सो नाउ there is one example I want to show. This is an interesting example that I always tell that, you know, sometimes mutation happens and we use mutation, natural mutation used for our crop improvement. For example, SD1 mutation, green revolution, all of you know green revolution. ठीक है, green revolution में हमने क्या किया था? Dwarf phenotype develop किया किस में? राइस में and wheat में. But that was molecular analysis was that this was because a deletion in SD1 gene, semi-dwarf gene, 383 base pair deletion in rice. So 383 base pair deletion happened naturally, and that causes dwarfing. And we have utilized the allele, mutated allele, for our own purpose to improve our other crops to make them dwarf. ठीक है, तो इसमें हमने क्या किया? Natural allele that disrupted a function by 383 base pair deletion naturally, उसका हमने utilize किया. But sometimes natural mutation can be wrong. For example, Oro's natural mutation. Oro's natural mutation क्या हुआ? G से A हो गया था. That causes the disease. That's a deleterious mutation. Similar, in case of plant also, sometimes due to natural mutation, the stop codon could arise at the middle or at the early position of the gene. Stop codon कहाँ पे रहना चाहिए? Where stop codon should be present? At the end of the coding sequence. But due to mutation, the stop codon, if it arises at the middle, then what would happen? Protein synthesis would be stopped there, and then you get a premature stop codon. This is called premature stop codon. And due to this premature stop codon, the whole length of protein would not be produced. The protein would be a truncated version, and this protein would not be functional. And that is the reason why some plants are blast disease susceptible. We tried to apply base editor whether we could correct this premature stop codon. So we applied ABE, and we have modified specifically this T to a C nucleotide, a red to a blue. So when T to C happened, then it becomes a CAG. CAG is not a stop signal. CAG is not a stop codon. It codes for glutamine. Now this protein revives its original form, and it goes until the end of the protein to produce the whole length of protein. And this is a resistant protein, so that it gives a blast resistance phenotype. Okay, this is one example. And then second one, this is I know I think most of you probably know this gene DST, drought and salinity tolerance, because already Vishwanathan Chinnaswamy sir's group has mutated this gene using CRISPR-Cas9, canonical CRISPR-Cas9, means knocking out. Okay, but if you read this paper, Huang et al. 2009, it's a genes and development paper. Here they say that this original natural DST mutant is due to N69D mutation. What does it mean? N means asparagine and D means aspartic acid. So asparagine से aspartic acid हुआ है, and that's the reason of mutation. Okay, and this mutation, this is the mutation for DST phenotype, drought and salinity tolerance phenotype. Okay, now yes, encouraged by the result published by Vishwanathan sir, yes, this gene is a good candidate. So I talked to him also, and then now we are trying to change whether we can mimic the same natural mutation in some of our cultivar that are very important. And if we mimic the natural mutation by changing asparagine to aspartic acid very specifically, can we also see, or hypothetically we should see, the phenotype drought and salinity tolerance. Okay, that was our hypothesis. So what was our target? N69 to D. It means if you go to the N69D is the protein level. If you go to the DNA level, N is coded by AAC. AAC is the codon for asparagine. Aspartic acid is GAC. Now what you need to make? A2, which base editor you would be using? Adenine base editor. Yes. So desired changes AAC to GAC. Let us see. We have tried this, and what you have seen, it's not as expected. This is the AAC codon. What do you see here? GGC. It became, why it became GGC? Bolo? It was very easy to answer. There is another A. Okay, डियामिनस को पता नहीं है कौन सा आपका टारगेट है, ठीक है, उसका ऑप्टिमम एक्टिविटी, इफ इट कम्स टू ऑप्टिमम एक्टिविटी विंडो दे विल इमीडिएटली डू दैट. Now this we have used the PKAV80NG because this target does not have NGGPAM, it has only NGPAM. Now you can also argue with me, sir ये क्लियर आप झूठ बोल रहा है, ये देखिए क्लियरली NGG है इधर है तो AG. But if it is an NGG, if you take this NGG, then this target A does not fall within the activity window, so it will go further. If you count, you have to count from here to here. Okay, so your target A would not be within the activity window. So बेस एडिटिंग गाइड के लिए आपको वो ख्याल रखना है आपका टारगेट कहाँ पे है, उस हिसाब से आपको एनजीजी ढूंढना पड़ेगा. उतना दूर में, तो उतना दूर में कोई एनजीजी नहीं था, बगैर एनजी था, इसलिए वी हैव यूज्ड AB80 NG version. But as I told you, AB80 is highly potent, very strong, and that's why all the 3A target A has been modified and it became a stretch of G. Now what modification we have done? We tried to do asparagine to aspartic acid, but we have got asparagine to glycine. So it is a noble allele that does not exist in the nature, but we have created and we have to phenotype them whether it is giving the resistant phenotype or not. That is need to be told only after experimentation. Okay, so almost I am finished with the base editing, but this last part I should tell you. So some of you probably interested in, genome is not only present in nucleus, it is also present in where? Chloroplast and mitochondria. But chloroplast, mitochondria are also the gene that is coded from the chloroplast and mitochondrial genome are also very, very important. Very important in the sense, for example, photosynthesis related, yield related genes, lot are present in plastid genome. So there are base editor has been developed using tail array. CRISPR-Cas9 is not suitable to edit any organelle genome. Can anybody tell why? Show why CRISPR-Cas9 is not suitable to edit an organelle genome? Partly true. Prokaryotic origin is obviously this good information, ठीक-ठाक information है. But why CRISPR-Cas9 is not able to edit mitochondrial plastid genome? Because you have to, what is the reagent for doing editing? Cas9 protein is a reagent and guide RNA. Okay. For example, for base editing, base editor is a protein and guide RNA. So यहाँ पे आपको प्रोटीन को भी ट्रांसपोर्ट करना है अंदर और आरएनए को भी ट्रांसपोर्ट कराना है. Yes or no? Protein can be transported into the organelle by using transport signal. For example, for mitochondria, if you want to transport any protein, you have to fuse the protein with mitochondrial transport signal, MTS. For if you want to transport any protein to chloroplast, you have to fuse a transit peptide TP. You see here is TP and MTS. Now protein can be transported, Cas9 can be transported, base editor can be transported, but there is no mechanism to transport guide RNA. We do not know. If you discover, you get a very good award. There is mechanism obviously, but people do not know what is the mechanism. That's why they cannot use them for engineering. Okay. So that's why Cas9 cannot be used. So that's why they have developed this base editor by fusing deaminase with tail protein. Tail does not. Tail is a protein. Tail can also specifically bind to any DNA sequence that you target, but you have to engineer the protein. But it does not depend on any RNA to bind. But Cas9 is dependent on RNA to bind to a specific DNA. But this protein can itself bind to a DNA. That's why it's a whole protein based. Tail is fused with deaminase and then it goes inside because it is fused with the transit signals and it can do editing. Okay. Now coming to second part. Take a breath because it was heavy, I know. Okay, tell me. Can you repeat your question, please? Oh, okay. So you see, common enzyme, for example, for it's very easy to know from SACO and digestion that I told you. Okay, so it's not that easy to select the target. So I spent at least 15 days to select that to target. Okay, so only restriction site is not also the only one criteria. The restriction site also should be in the optimum activity window of the base editor. So that was lot of search and then I could find this too. So for you, base editing experiment does not have lot of option for you to change your guide RNA. Normally you have to change this, you have to use the single guide RNA. You do not have to use the software to design guide RNA for base editor. Because for example, DST target, N69D is our target. So N69 means 69 is the position amino acid which is crucial. You do not have any other chances. You have to target that one. You have to choose the best guide RNA from that region where the asparagine falls within the activity window. For example, for our case, the DST case, there was no restriction enzyme. Have I showed you any restriction digestion experiment? No. But what you have to do, you only have to do a Sanger sequencing to evaluate your editing. There is no chance of easy genotyping. Got my point? That is the only option, yes. Okay? So you are saying most of the protein are synthesized. We cannot edit protein. But if the gene is coded from chloroplast, you have to edit there. Have you got my point? If the gene is coded in nucleus, there are a lot of protein that acts within the chloroplast but coded from nuclear genome. You can do that kind of editing, no problem. But got your answer? Okay. Now go to the next problem. What is this problem? We are trying to do increasing nitrogen use efficiency in one of the gene that needs TC2AA change. If we can do TC2AA change, we can inhibit a microRNA binding site. So this gene is inhibited by a microRNA. but there is one natural mutation found, and that is high in nitrogen use efficiency, and that natural mutation was found as TC is modified to AA nucleotide. So it means TC, when it becomes AA, the gene become microRNA resistant. So gene mRNA would not be destroyed by the microRNA cleavage. Okay, we wanted to mimic this, but do you think it is possible to do it with base editor? जल्दी बोलो, is it possible to do it base editor? हाँ, एक साथ दोनों करना है, एक T एक C. Yes, you can change C to T, but you cannot change T to A. It's not, there is no base editor. So how would you do that? For example, this is another example from animal perspective. This is a Tay-Sachs disease, and this disease, the children mostly die by the age of 5. This is an inherited disease, and the reason is in hexa gene 4 extra base pair is inserted in the diseased children. Diseased children, they are suffered from this disease because they have 4 extra nucleotide inserted in the hexa gene. Now if I ask you what is the hypothesis you want to cure this baby, क्या करना है आपको? जल्दी बोलो. Delete that extra 4. These wrong 4 base pair insertions should be deleted in order to correct the gene, okay, and then the disease phenotype would also be corrected as a, this is a hypothesis. Now it is not possible. Can we do that with canonical CRISPR-Cas9? Okay, मुझे पता है CRISPR-Cas9 से deletion होता है. कितना base pair deletion है आपको पता होगा? That's not in your hand, that's a random case. Okay, so you wanted to change 4 base pair deletion, you wanted to do 4 base pair deletion, it can achieve 7 base pair deletion. Then the gene is further broken, you want to repair the broken gene, now you have further broke the gene. So that's not expected. Okay, for example, another changes to here from our laboratory that we are working on a project on C4. So if you see, the PEPC gene is a C4 photosynthetic gene, all of you know PEP carboxylase, but those C4 photosynthetic gene is also present in C3 rice, and there are 2 amino acid changes when we compare C4 and C3. This is the amino acid changes. In C3 arginine is present, in all C4 glycine is present. What does it mean? This arginine to glycine transition and here alanine to serine transition might have some role in C3 to C4 evolution. So this paper says that this alanine to serine conversion in nature is responsible for the higher catalytic activity of C4 PEPC gene, and these arginine R to G is responsible for lower malate sensitivity. PEPC is an enzyme. This enzyme can be inhibited by malate, and if there is glycine, it is less sensitive to malate. If it is arginine, more sensitive to malate. So it means these 2 amino acids are very, very crucial for making a C3 PEPC to a C4 like PEPC. Am I right or wrong? ठीक है, तो जो उतना क्रूशियली मत जाओ. सपोज ये दोनों हमारा टारगेट है. ये दो, in order to achieve these 2 changes, one change is required, G to T modification, for another C to G modification is needed, but the base editor cannot be applied here. When you have analyzed the DNA, base editor is not applicable. So these all 3 cases, one case is nitrogen use efficiency TC to AA, and then hexa gene, the Tay-Sachs disease, and then this one, base editor is not applicable. One thing could be applicable. What is the technology? No, prime editing I have not introduced you. जो हमारा presentation से पता है वो कौन सा technology? Yes, HDR is the only technology that you can do. You can apply to do this kind of all edits. But HDR is 0.01. So आपको 10,000 generate करने से 2-3 मिल सकता है, ठीक है, 4-5 मिल सकता है. So it is very difficult in our situation. But this technology is called search and replace genome editing technology. That can achieve all 12 types of base substitution, insertion, deletion, and combination of above. Remember, this is the range that I mentioned here. This is reported in the first initial original paper by Angela et al. That's why I mentioned here. But now prime editing advanced to a higher level. So all kind of editing, complex editing you can do with prime editing. This prime editing is like a word processor. You can delete, you can insert, you can replace, you can do combination. Suppose you have made a project report and you submit it to your supervisor, and supervisor said, hey, this word is not suitable here, okay, replace with this word. So whole word is being changed. Now another teacher said, no, this letter is not good, change this letter, okay, in order to refine your draft, okay. That can be done similarly in the genome by using prime editing. What is it? The NAHEP-CAAST is fused with a reverse transcriptase enzyme. NAHEP-CAAST fused with a reverse transcriptase enzyme, not deaminase. And another difference is here, this is a normal guide RNA, but for example, this normal guide RNA is all true for base editing and canonical CRISPR-Cas9, but for prime editing you need to use a modified guide RNA, that's called PEG RNA, prime editing guide RNA, which has an extra sequence here. And this sequence is nothing but this is called search sequence and replace sequence. This region is search region. Search region means this will direct where to edit, and this region will direct what to edit. Got my point? Where to edit, this region, what to edit. So the same guide RNA, PEG RNA, is now coding for two different functions. One is कहाँ पे जाना है और क्या करना है. ठीक है? Clear? How it works? Problem? 3 base pair wrong है. 3 nucleotides is wrong. You have prime editing. आप 3 base pair को delete करके क्या कर रहे हो? 1 nucleotide insert and then you get your phenotype back. So prime editing is working in this fashion in the genome. Now if you see this, this is how it works. This region is what? बोलो नीचे जो रहता है क्या है? Search means आपका same region जो आप normal CRISPR-Cas9 में डिजाइन करते हो 20 base pair, this is the same region. Search region and then this is the replace region. इसमें दो चीज़ रहता है, एक primer binding site, another is RT template. Primer binding site is the same, you cannot modify that. That will make a pair with a nicked strand. Why there would be a nick? Jaldi bolo. Prime editing is made up of N-Cas9. So there would be only one nick in this strand and there is one flap will be moving around. With that, this prime editing site will bind with whole homology, and then here is the region where you have put intentionally your changes that you want to bring in the genome. Got my point? So ये RT टेम्पलेट में आपको डिज़ाइन करना है, आपको क्या चाहिए, आपको TC से AA चाहिए या हेक्सा को 4 बेस पर डिलीशन चाहिए या C2G चाहिए, जो भी चाहिए आपको मॉडिफाई करना है RT टेम्पलेट में. And then this gray color is the N-Cas9 and this what is this yellow color is the, suppose for example reverse transcriptase. I have shown here in for this animating this one. This is the reverse transcriptase, the violet one, and it will read the RT template and directly write what is written in RT template. It will directly write to the nicked strand. The black color strand is a nicked genome, okay? Black is genome, no? So it will directly write opposite to A, it will write what? Opposite to A, T. Opposite to U, A. Opposite to C, G. It will directly write in the nicked genome. So it's a direct writing of your intended changes into the genome. This is how prime editing works. Am I clear or not? आपको अगर ज़्यादा complexity से पढ़ना है, आप घर में जाके बहुत अच्छे से पढ़ सकते हो, but this is the basic things to explain without making you too much boring. ठीक है? Okay? Yes.
How do we decide where should our
Exactly. Yes. So there is earlier.
tools available?
Yes. But what I should say earlier, when we designed our first experiment, seeing the 2019 paper from David Liu, that time we designed manually. Okay. Only the recommendation that they have given in the paper, and they have said, okay, 13 base pair RT template. We recommend 13 base pair RT template. We recommend PBS site should be 9 or 10 base pair. So we followed their instruction, okay. But now there are huge changes in the technology. So new software already came, and they consider your genomic context also, and they will rank RT. Yeah, I'll tell you. I have that. I have that slide. Okay. Okay, so now it is very easy, because designing a prime editing guide RNA is really tricky if you do manually. But remember, if you learn manually, you'd be an expert. Because we learned primer designing manually, we learned handling sequence manually in our PhD. So that's why when we use software, so we are double strength than you, those who only use software. Okay, so if you learn manually fast and then go to software, you can even detect some error from the software also. Okay, so using that manual designing, okay. So we failed to achieve any edits in one or two of our targets. 300 plants regenerated, nothing happened. And other group also reported that PE efficiency is really low in plant. But now groups are also started reporting from plant scientist fraternity that the prime editing efficiency is likely increasing in a very good rate. So well, we immediately started to improve the prime editing efficiency by two different approaches. One is, suppose this is a PEG RNA, and where is the replace section? जल्दी बोलो कौन सा एंड में है? 3 प्राइम एंड में रिप्लेस सेक्शन होता है, एंड रिप्लेस सेक्शन में आपका क्या है? which is being coded from the replace section, the edit that you want to make. But a search section is where? Here in the 5 prime end. But remember, when Cas9 and this guide RNA make a complex, up to these, the Cas9 protein actually masks the whole RNA. So it is not exposed to the cellular nucleus. But this region, this extra 3 prime extension, is exposed to the cellular environment, and cellular environment has lot of nucleases, and they can degrade the 3 prime end. That's why we wanted to fuse something that will inhibit the attack from any nucleus, and that's an RNA aptamer to prevent degradation. This is one approach. Another approach, this is a bit further complex mechanism. So to improve prime editing efficiency, we also fused one human repair protein that is likely to, as our hypothesis, likely to increase the efficiency of prime editing. These are two approaches we are trying. So when we did, we have designed 5 different prime editing constructs based on the initial construct by fusing and modifying the guide RNA, and you can see here is P1, P2, P3, P4, P5, and there are 2 target 1 and target 2. T1 is target 1, T2 is target 2, and you can see there is a gradual increase. These are all validated in protoplast. This is a gradual increase in editing efficiency. And what you see here intended, for example, here the example I put, we wanted to achieve the C2G editing in the Pepsi gene, C2G. So what you see, this is the C target and it becomes C. How many percent is here? 4.89%. So 4.89%, it is, at that time it is really exciting. 4.89% means if you generate 100 plants, how many plants you will get? At least 4 you would get. If you are lucky, you would get 7, even 8, because that time division will happen, okay, cell division will happen. So it's a good encouraging result. Then we transfer, with our best vector, we already did callus transformation, and this is a recent data. You can see C2G happened in 48%. That is a huge achievement, and we are really excited with this data. And you can see there is also, you should know that prime editing is not too precise, because what are the other outcomes? Can you just measure? You have C2G, targeted changes happened, but what other things happened? Dot means? So your desired change is there, unintended change is also there. Lot of cases are there where your desired changes is also there, unintended changes is also there. So you only need which one? Only the desired one. So you have to figure out how many percentage of pure product you are getting. So this is called pure editing, is 48.5%. And now you see when plant is regenerated, we analyze them, and you can see there is a 2 example. This C, the target C has been modified. C means blue to black. C2G has been already achieved in that endogenous Pepsi gene in our target. Okay. So this is the story of prime editing, and this is the answer of madam. There is huge list in this paper, but I am not recommending you, because otherwise you would say that Kutub is very selfish, is not saying the best. So we use plant peg designer, is developed from Teja Gao from China. Plant peg designer, and another is, here is plant peg designer at the bottom. So you can ask why you have kept plant peg designer at the bottom, because that when we reviewed, plant peg designer was new that time. So it's a single order. And then another is peg finder. These two I would recommend, peg finder and plant peg designer. What we do, we use both of them and then we cross check whether both of them is getting the same output. If they are giving the same output, then we are super confident, but both the software is recommending us the same. Okay, clear? So now these are few advanced things. You can now achieve huge 10 kb के आसपास भी डिलीट कर सकते हो. दिस इज कॉल्ड डुअल पेग आरएनए. अब दो पेग आरएनए यूज कर सकते हो, इधर एक ऊपर स्ट्रैंड में एक, नीचे स्ट्रैंड में एक. 2 peg RNA simultaneously you would add, and their replace search is different. One is here search component, another is here search component, but their replace component is same. Got my point? 2 peg RNA we are using, that's called dual peg RNA, and this 2 peg RNA's search component is different, but their replace component is same. And these are huge, these are all prime editors, different prime editors recently developed, that can give you lot of different huge level of editing, like 10 kb deletion, 9 kb deletion they have achieved. Yeah. Exactly, in both sides, yeah, that's a limitation. Yes, you are right. In both sides you have to have NGG. Yes, exactly, you are right. So that's a limitation. Yeah. And then wild type, recently people have also developed wild type Cas9 use करके वो prime editing develop किया. So they have used, instead of N-Cas9 they have used wild type Cas9 to develop prime editing, and it's normally for deleting. It's not for other. Even I am now reviewing one paper. You know, Trends in Biotechnology is actually now started publishing original paper. For your information, Trends in Biotechnology is a review journal. Okay, now it already started publishing original papers. So there is one development from China also. They have developed prime editing for plant using wild type Cas9. Okay, so I am at the end. I have only 2 to 3 slides more. Yes, sure. Yeah, sure.
So, when we did it, there was this newspaper had come. So we started off with different targets, and selfishly we chose a target which was already edited using taggers.
Yeah.
So to just check whether as a proof of concept. That worked, but the new target did not using primary. And we got a 44 base pair insertion also.
Yeah, that's.
That was again a checked site. So does that mean that there is a trade-off between screening when we are using some site which is already being used versus something which is absolutely under, because probably it's not accessible. We have to screen a huge population. Have you had an experience with an absolutely new target?
No, absolutely new target has a lot of different explanation. For example, you know, genomic context also very important. So there is one new tool recently developed for mammalian system. You are excited to see that, because they consider whether the genomic context is already, the genomic region that you are targeting is already epigenetically modified, it is methylated or not. If it is in heterochromatin region also, you do not know the status of your gene, where it is lying. So that is probably the reason, and you have chosen already talent validated guide, and that work means, yeah, exactly, because it.
Can we screen larger and hope? That's my question. Or no?
That, yes. I think it could be possible also. If you screen large, you can get 1 or 2 plants. Yeah. Okay. Thank you, madam. Okay. So thank you for joining. Both of you, I know, director of 2 institutes. Yeah, last I have 2 or 3 slides more only to tell you what kind of application could be done, and these are all demonstrated applications. It's not potential applications. For example, herbicide tolerance, B and P, base editor and prime editor, has already been used to develop herbicide tolerance. in those plants, and then also altering nutrient composition, like altering starch composition and also sugar composition, lycopene content. And rapid domestication also in Oryza alta, they have used base editor to do rapid domestication, especially to modify one or two wild characters to make them like domesticated one. Yield improvement obviously, and then disease resistance. I showed you already example from my cases. One inferior allele is modified to a superior allele, so it's a susceptible phenotype to resistant phenotype. And then input use efficiency also. And just to show you, because just yesterday I was talking and they said you must present this work at least 2 to 3 slides. So it's not related to base editing or prime editing, but we have recently came up with this alternative of Cas9 and Cas12. If Cas9 is a football or soccer ball, our tool is only 408 amino acid. It's only a baseball. It's 1/3 of the size of a Cas9 or Cas12. So we have successfully demonstrated in rice and Arabidopsis, and you can see in one cases the mutation we have achieved up to 69%, and we have demonstrated multiplexing with this tool and demonstrated in Arabidopsis. Also we have developed base editor with using this TNPB, and also we have constructed TNPB mediated gene activator. So I am not going to tell you, but I would recommend, if you are interested, you could read that paper. And these are all possible to rapidly do that because of our established protoplast system in rice that we recently established in our laboratory. And you can see we targeted 2 very easily scoreable phenotype governing genes. One is HMBB. Both are related to chlorophyll biosynthesis. And we achieved 53 base pair deletion and 23 base pair deletion. You can see the albino phenotype here with that small size genome editor. So I'd finish with this video because I started with the which example? Progeria. I'll finish.
See if I can. She'll let me pick them up. Hi, honey. Hi. There he is. Let me see everybody. Say hi. And then we have, as compared to the C57, they still have a very nice sleek coat. They're still very, very active. If I were to pick them up, they can jump out of my hand quite literally.
So you got my point? So it's already validated in mice. By using base editor, they have cured a progeria affected phenotype. Okay, thank you. So if you want to apply the experiment to your own crops or systems, these are the 2 protocol books that we have edited. You could go through and just, they have all troubleshooting and material methods and stepwise protocols. And I must acknowledge that I learned genome engineering from my PhD life from Shapun Dotto and Kaurabi Dotto madam. And then in postdoc with Professor Inong Yang, I learned genome editing in his laboratory at Penn State. And this is my current group, who is really, really very, very hardworking. Means I cannot tell you how much hardworking. They go back at 9 p.m. and then they cook food, sir. So they are really very hardworking. And these are few photos to encourage all the students. These are with tiny follower with pioneers. So this is James Watson, Jennifer Dorner, CRISPR-Cas9, and the talent pioneer Dan Vartas, and this is for base and prime editing David Liu. Thank you very much. Thank you. Okay, sir.
So thank you so much, Tutab. Since our next speaker is already here, so we will not take any question right now. But Tutab is around. If any of you have any question, you can interact. But before that, we would like to felicitate Dr. Tutab. For that, I request Dr. Bhattacharya to do the honors. So friends, it's a matter of immense pleasure that our next speaker is Professor Ashwini Parith. Professor Parith is Executive Director at NABI Mohali, and he is also Professor at JNU, SLS JNU, and Adjunct Professor at University of Western Australia, Perth, Australia. Uh, Professor Parith is established, very well recognized plant biotechnologist. He is fellow of almost all the academies of India, all the scientific academies of India, and I thank him for agreeing to our request. Please, sir.
Thank you, Anil.
सर ये वाला है ना?
Okay, while our experts are setting it up, let me take this pleasure and honor to say thanks to the organizers. It's always been a pleasure to come here to NAHEP and interact with friends and colleagues over here, Dr. Bhattacharya, Anil Singh, and many other. Thank you. And I also got a chance to listen to Kutub's presentation. Thank you very much. We all know that gene editing is coming up in a big way. This is mine. And all of us know the importance of using such kind of pseudogene technologies. We all set? Yeah. So I'm left with roughly 4 is that how can we make use